All right, good afternoon, everyone. Thanks so much for the opportunity to share this project that I've been working on. So this is me. I'm actually a pediatrician and a nephrologist. I have an active clinical practice, but I'm an investigator in the Vascular Biology and Therapeutics program at Yale. My area of focus is vascular inflammation, steroid microenvironments, and endothelial cells, and more recently, Wnt signaling. The problem that I'm trying to tackle is that of cardiovascular disease, which remains the #1 cause of worldwide mortality. Despite many FDA-approved drugs since 2010, mortality continues to increase, and healthcare costs are also increasing in the range of hundreds of billions of dollars. So clearly the approaches that we have are not sufficient. Patients with cardiovascular disease have a lot of homework, a lot of modifiable lifestyle factors that they're supposed to address. And besides that, the pharmacotherapy is mainly centered around lipid-lowering agents. Our lab has identified the Wnt signaling pathway as a key mechanistic pathway in the development of cardiovascular disease through an unbiased genomic screen. However, there are no current therapies for cardiovascular disease which address this pathway. So here I want to share some data that we've generated in my laboratory. So I'm showing here isolated mouse aortas stained with oil red O, which turns red wherever there is lipid. And these mice were fed for 12 weeks with a high-fat diet, and after 4 weeks of diet, they were given a Wnt inhibitor for 8 weeks. I have two models here. One is the ApoE knockout model on the left. More. Oh, hello? Oh, much better. Okay. Hopefully you heard some of what I said. So anyway, yes, these are isolated mouse aortas that were dissected out from animals fed a high-fat diet for 12 weeks. After the first 4 weeks, they received a Wnt inhibitor, which is termed here LGK974. And there are two models here. The first is the ApoE knockout model, which is a standard model of mouse atherosclerosis. The second model is the model I generated, which is called the DKO model, which is more severe, obviously, you can see. And it's a knockout of the endothelial glucocorticoid receptor on the background of the ApoE knockout animal. And you may be wondering, why did we even attempt to use Wnt inhibitors in this model? And that was because when we were trying to understand the severity of the phenotype, we did ChIP-seq for GR and endothelial cells and demonstrated that in the absence of endothelial GR, Wnt signaling was highly upregulated. So basically, you can see with the addition of the Wnt inhibitor, the lipid deposition is decreased by about 50%. We can also demonstrate a significant lipid lowering effect of about 50%, which is similar to standard of care and perhaps better. However, we can also demonstrate two additional mechanistic insights, which are not addressed in current therapies. The first is mitigation of inflammatory cytokines. Excuse me, my voice is very bad after talking during lunch. The first is mitigation of inflammatory cytokines, and you can see the addition of LGK974 suppresses inflammation by about 50% in the blood. And secondly, we can show in isolated endothelial cells that there's restoration of fatty acid oxidation, which is very important because defects in fuel preference have been shown to contribute to fibrosis and mortality. So currently I've been awarded about $3 million in NIH funding to study this process. Currently I have a patent that was issued last June. The claim set allows for the use of Wnt inhibitors as therapy for cardiovascular disease, and it allows for new compositions of matter. So this would represent a first-in-class therapeutic opportunity. And basically what I would like to do is develop an endothelial-specific Wnt inhibitor by doing a high-throughput microscopic screen in endothelial cells using several curated small molecule libraries which have been selected based on the mechanism of action of the current inhibitor, and then eventually take those leads into our validated mouse models and do comparative efficacy studies based on current standards of care. Thank you.
Do you have questions from the audience?
So for the phase 1 use, what is it about the endothelial cell types that would lend itself to some specificity for these libraries of molecules you'll be screening, Wnt and endothelial versus other cell types?
Well, I think it's not that the cell type would necessarily lend itself very well, but the fact that endothelial dysfunction is generally recognized as the first step in the development of atherosclerosis, so it would be the very earliest intervention phase, and also the fact that many of the beneficial effects we've seen with the inhibitors can be mediated solely through endothelial cells.
But these would be administered systemically, hit all the cells.
Potentially, I think one part of the development could be demonstrating the specificity to endothelial cells, and if that was not the case, there are endothelial-specific drug delivery platforms, which I think could be employed.
Great, thank you very much.
Thank you.